Welcome to another session. In this session, we're going to learn about transport across cell membrane. So there are three different types of transport mechanisms: passive, active, and then vesicular transport. In this session, we're going to mainly focus on passive transport, and in the next sessions, we will learn about active transport and vesicular transport. So, what is the importance of this topic, which is transport across cell membrane? So the topic itself is explaining that there are some substances which are transported across the cell membrane. Why it is very important? So the physiological activities of the cells depends upon the substances like nutrients, oxygen and water, which must be transported into the cell, and at the same time metabolic waste products and the end products must be removed from the cell. So these waste products must be transported out of the cell and the nutrients must be transported into the cell. So the cells achieve these by means of different transport mechanisms across the cell membrane. So these transport mechanisms are classified into passive transport, active transport and vesicular transport. I told you we are going to focus mainly on passive transport. So what is mean by passive transport? So transport of substances along the gradient without expenditure of energy, that is a definition for passive transport. It is self-explanatory. Passive, which means no energy is needed and it is along the gradient. So transport of substances along the gradient without expenditure of any energy is called as passive transport. It depends on the factors like concentration gradient, electrical gradient and pressure gradient. So these here we can see in the passive transport, the substances are transported from higher concentration to the lower concentration. And these two are the different types of passive transport mechanisms. Here you can see diffusion and facilitated diffusion. These two are the different passive transport mechanisms. In both of these, you can see the substances from the higher concentration, they are transported to the lower concentration. So it not only depends on concentration gradient, it also depends on electrical gradient and pressure gradient. So it is a downhill movement because it is transported from higher concentration to the lower concentration. So it is also known as downhill movement. So you must remember these points. Passive transport is transport of substances from the higher concentration to the lower concentration, that is along the gradient without expenditure of any energy, and it is also called as downhill movement. So here you can see the how the passive transport, what are the different types of passive transport mechanisms. So passive transport is classified into diffusion and osmosis, and diffusion is again two types: simple diffusion and facilitated diffusions. We will see one by one. So we know what is passive transport. Passive transport is along the gradient without expenditure of energy, and it is a downhill movement. So what is diffusion? What is diffusion? And diffusion is nothing but it is the passive transport of molecule from the area of higher concentration to the lower concentration. And this diffusion, I told you, it is of two types: simple diffusion and facilitated diffusion. First we will focus on simple diffusion. So here, simply through the cell membrane, from the higher concentration, the substances are transported to the lower concentration area. So this is called simple diffusion, simply through the cell membrane. So any molecule which is lipid soluble, as you know, we studied in the cell membrane, the structure of cell membrane. Cell membrane is a lipid bilayer membrane. So and it is a semi-permeable membrane, you know that. So the substances which are lipid soluble can easily pass through this cell membrane by this simple diffusion. So what are all the examples we can say? Oxygen, carbon dioxide, nitrogen, and there are many other molecules like steroid, alcohol, weak acids and bases. All these are being lipid soluble, they are easily can pass through the cell membrane, and these are the substances which are easily passed through the simple diffusion. And this simple diffusion is mainly due to the random movement of the molecules. Here we can see there are more number of molecules. Because of the random movement of molecules, they collide with each other and the molecules are transported from the higher concentration to the lower concentration. And this diffusion will occur till the equilibrium reaches on both the sides, and this equilibrium is known as diffusional equilibrium. So Fick's law of diffusion explains about the simple diffusion, where J is the rate of diffusion and the rate of diffusion is directly proportional to the concentration difference. You can see C1 minus C2, that is the concentration difference of the substance, and A is the cross-sectional area. A is the area, which is cross-sectional area, and that is surface area, and T is the thickness of the membrane. As you know, rate of diffusion is directly proportional to the concentration difference and surface area, and it is inversely related with the thickness of the membrane. And there are many other factors which is related to this Fick's law of diffusion. It is not only depends on the diffusion is not only depends on these factors. There are many other differences. Just remember, according to Fick's law of diffusion, rate of diffusion is directly proportional with the concentration difference and the surface area, whereas it is inversely related with the thickness of the membrane, which means what? If the thickness is more, then the diffusion is less. So that is the meaning for this. And if the concentration difference is more, then the diffusion will occur more. And if surface area or cross-sectional area is more, then the diffusion is also more. So that is the meaning for this. And what are all the factors which affects this diffusion? I told you, so being lipid soluble substances will be, they will have high diffusion through the cell membrane. So lipid solubility, the lipid soluble substances are directly proportional with the diffusion. They water soluble substances and the substances which are lipid insoluble substances, they have inverse relationship with the diffusion. The other factors like type of electrical charge and the size of the molecules and the selective permeability of the cell membrane, all this can also affect cell membrane. What are the other factors which are according to Fick's law of diffusion, even the thickness of the membrane is inversely relational with the diffusion, whereas the distribution of the protein channels, we are going to study what is protein channels, and the number of protein channels can also affect this diffusion. And one more thing is temperature. The rate of diffusion increases with increase in the temperature. And the size of the molecules. Size of the molecules, as you know, if the molecules are larger, then the diffusion will be lesser. So it is also have a inverse relationship with the rate of diffusion. So all these are the factors which affects diffusion. And lipid soluble substances, they easily pass through the simple diffusion. This we discussed already. So rate of diffusion is directly proportional to the lipid soluble substances. Examples like oxygen, nitrogen, carbon dioxide, alcohol, steroid hormones, weak organic acids and bases, all these are lipid soluble substances which can easily pass through the cell membrane. Through simple diffusion, these substances can pass from higher concentration to the lower concentration without expenditure of energy, that is along the gradient. So this is what we should remember. And even the water soluble substances and lipid insoluble substances can also pass through the simple diffusion, but with one condition, that means the cell membrane should have some protein channels. They are called channel proteins. Already we studied that there are three different types of proteins which are present in the cell membrane. One type is the integral protein, which is also called as transmembrane proteins. I told you they will pass through and through the cell membrane. So here you can see these transmembrane proteins, they are passing through the cell membrane, and these are called channel proteins. And these channel proteins help in the transport of molecules. So that is the lipid insoluble substances and the water soluble substances can also pass easily through this channel proteins. But the channel proteins, mainly the permeability of the substances through these channel proteins depends on the molecular size, shape and even the charge of the substance. So these channel proteins, they have two important characteristic features. One is selective permeability, the other one is gating mechanism. What is meant by selective permeability? These channel proteins, they will allow only one type of molecule. For example, if we say sodium channel, then sodium ions can pass through the cell membrane. It will not allow any other ions. If it is a potassium channel, it will allow only potassium ions to pass through the cell membrane. It will not allow any other ions to pass through the cell membrane. So that is the meaning for selective permeability. And the second important feature of the channel proteins are it has a gating mechanism. In some channels are always opened, whereas some channels will have gates like extension. These are tube-like extension which are present in the cell membrane. These are tube-like extension which are present in the cell membrane. Here you can see there are two examples which is shown as a gated channels. So here we have sodium gated channel, and here we have potassium gated channels. So the sodium gated channels have gates like extension on the extracellular fluid. It has on the outside we can see the sodium gate like extensions are present, whereas in the potassium channel the gate extensions are present in the intracellular fluid. So the very important feature about this channels are, these are the actually the examples for voltage gated channels. Gating mechanism, according to gating mechanism, according to the opening or closing of the gating channels, these gating channels are classified into three different types: voltage gated calcium channels, ligand gated gating channels, and then there is a third type which is called as mechanical gated gated channels. And what is the meaning for voltage gated channels? Which means they depend for their opening, they depends on the change in the voltage. For example, here we have voltage gated sodium channels. The sodium channels are closed when the interior of the cell membrane is negative. When the interior of the cell membrane is negative, these sodium channels are closed, whereas when the interior of the cell membrane becomes less negative, you can see here there is more negative and here we have less negative. So whenever there is a negativity decreases inside the membrane, these sodium channels open, the gates of the sodium channels open, and there is sodium influx occurs. You all know this, and we are going to study in the later classes there that the action potential or the depolarization state of the action potential mainly depends on the sodium influx of the sodium influx through the cell membrane. So this is one of the examples. So sodium influx is the reason for the generation of action potential, generation of nerve impulses across in the cell membrane, and this is due to the opening of the voltage gated sodium channels. So we can see one more example, that is the voltage gated potassium channels, and here the gates are present inside the cell membrane, and these gates will open whenever there is increase in the positivity in the cell membrane. So inside of the cell membrane, if it is negative, these potassium channels are kept closed. When the inside of the cell membrane becomes more positive, then these potassium channels will open, and that will cause efflux of the potassium, because these gates are present inside the cell membrane. When these gates open, potassium efflux will occur, and this is the reason why there is repolarization phase of the action potential occur. So depolarization is due to sodium influx into the cell membrane, which makes the interior of the cell membrane becomes positive, and that is a state which is called as depolarization. And during repolarization, potassium efflux occurs, and because of this potassium efflux, inside of the cell membrane becomes more negative, which is going to again to the polarized state, repolarization, which means it is going to become again negative. So the interior of the cell membrane is going to become negative by decreasing the positive ions. That is mean the positive ions, that is the potassium ions are going outside. So that is the potassium efflux. So this is very important to understand because this will help you in the later classes when we study nerve muscle physiology to understand the action potential and the resting membrane potential. So this is one type of gating mechanism. This is especially voltage gated channels, because they depends on they'll open or close based on the voltage on the membrane. And next type of channel is ligand gated channels, and here also we have the voltage gated sodium channels only, but ligand gated channels they'll open when some ligand, which means the ligand means the chemical molecule. So whenever some a chemical molecule binds with these channels, these gates will open. And the third type, mechanically gated channels, when there is a mechanical stretch, these protein channels will open, and this is called the gating mechanism. So channel proteins will have two important features. One is selective permeability. It will allow only one ions to pass through, and the second mechanism is gating mechanism. And this is about simple diffusion. So simple diffusion is nothing but it's a type of passive transport. It is along the gradient, and it is without expenditure of energy. And simple diffusion can occur easily through the lipid soluble substances. Even the lipid insoluble substances and water soluble substances can pass through simple diffusion, but with the help of channel proteins. And these channel proteins have two important features. One is selective permeability, the other one is gating mechanism. And we should also remember about while writing about the simple diffusion, we should not forget about the Fick's law of diffusion, where the rate of diffusion is directly proportional with the cross-sectional area and the concentration difference, and it is inversely relation with the thickness of the membrane. So coming to the next type of diffusion, which is again a passive transport mechanism, facilitated diffusion. What is the meaning for facilitated? There should be some facilitator like me who should push you. So there should be some facilitator. So here we have some protein which is called as carrier protein. This helps in the transport of molecules from higher concentration to the lower concentration. So that is why facilitated diffusion is also called as carrier mediated protein, carrier mediated diffusion. So here we need carrier proteins on the cell membrane, and these carrier proteins helps in the diffusion of molecules from the high concentration to the lower concentration, and it is also along the gradient, and it is also without expenditure of energy, that is through the carrier proteins. And the examples of substances, we have simple diffusion. Why? There is one more type of transport mechanism. There are some substances which cannot pass through the simple diffusion, and those substances like glucose and amino acids, they are transported through this mechanism, that is through they'll bind with the carrier proteins, and these substances will be transported. So the examples for substances which are transported through facilitated diffusion is glucose and amino acids. This is very important point to remember, because there are many MCQs which can be asked from this topic, and glucose and amino acids are the substances which are transported through facilitated diffusion. So the mechanism of facilitated diffusion, how the substances are transported from through these carrier proteins. So whenever here you can see there are carrier proteins in the cell membrane, and these carrier proteins will have binding areas. So binding sites will be there in these carrier proteins, and here you can see some molecule will come and bind with the binding site, and the conformational change will occur in the carrier protein, and this conformational change will help in the transport of molecule into the cell. So this conformational changes will occur repetitively, and the molecule will diffuse from the area of higher concentration to the lower concentration. So this is how the facilitated diffusion will occur. And there are three different types of carrier proteins. So we told channel protein. We talked about channel protein in the diffusion. Here we are talking about facilitated diffusion. Here we are talking about carrier proteins, and there are three different types of carrier protein systems. So uniport, this is symport, and this is antiport. And symport and antiport both are together called as co-transport mechanism. Symport and antiport, they are also called as co-transport mechanism. Uniport, what is mean by uniport? Uniport, which means only one substance, right? So here there is transport of only one substance through this carrier protein. So that is the type of transport mechanism which is called as uniport. And the second type of mechanism which is called as symport. What is the meaning for symport? Which means there is some other substances. So the transport of one substance will be linked with the transport of another substances. So that is the meaning for symport. The best example for this is whenever sodium goes, along with that it will also take glucose, especially this happens in the intestinal cells as well as in the renal tubular epithelial cells. So that is the example for this symport. So along with sodium, the glucose will also transported in the renal tubular epithelial cells. That is the example for symport. And antiport, so the transport of substances in one into the cell, and there is one, the substance will be transported in opposite direction. The other substance will be transported in opposite direction. So antiport, which means one substance will be transported into the cell and the other substance will be transported out of the cell with the help of same carrier protein. That is a very important point to remember. With the help of same carrier protein, one substances will be transported in one direction, the other substance will be transported in opposite direction. So that is the type of transport which is called as antiport. And we can say sodium hydrogen exchange in the cell membrane, potassium hydrogen exchange in the cell membrane, which occurs usually in the renal tubular epithelial cells. And these are the examples for antiport mechanisms. So coming to the differences between the simple and facilitated diffusion mechanisms. So both of these are passive transport mechanism. So both do not require energy, and both are downhill movement. You know, they both are from high concentration to the lower concentration. And if you take simple diffusion, the channel protein will have specificity. So it is the specificity is not there for simple diffusion, whereas facilitated diffusion, it is specific for some substances. And carrier proteins. Simple diffusion, if in case of simple diffusion, the channel proteins they will allow any type of molecule, but facilitated diffusion, it will allow only some specific molecules. And if you take carrier proteins in the simple diffusion, it is not required. Here we need channel proteins. In facilitated diffusion, we need carrier proteins. So this is carrier protein dependent mechanism. Examples: oxygen, carbon dioxide, nitrogen. So lipid soluble substances can pass through this, can pass through simple diffusion. Whereas facilitated diffusion, glucose and amino acids, these are the best examples for the substances which are transported through facilitated diffusion mechanism. Speed of diffusion, here it will be slower and here it will be faster. And the very important point, which is saturation, we'll see with one graph. These 2 points will I'll explain you with the graph, which is very important for exam purpose, and it is also very important to understand that graph to understand these points. So rate of diffusion increases proportionately with the increase in the concentration of substance, and there is no limit to it. This happens in simple diffusion. Whereas in facilitated diffusion, rate of diffusion increases with the increase in concentration gradient to reach a limit beyond with a further increase in the diffusion cannot occur. This is called saturation point. What is this? We will explain with the graph. You will understand this clearly. And here you can see this is graph A and this is B. And here you can see rate of diffusion and then the concentration of the substance. Just remember, whenever in simple diffusion, whenever there is increase in the concentration of the substance, there is increase in the rate of diffusion. So whenever there is increase in the concentration of substance, rate of diffusion also increases linearly with the increase in the concentration of substances. This happens in simple diffusion. But whereas when you take facilitated diffusion, the rate of diffusion increases as the concentration of substance increases, but it is only up to a certain point, and that point is called saturation point. After this, even though there is increase in the concentration of substance, there is no increase in the rate of diffusion. So this saturation, there is a saturation point for facilitated diffusion. It is mainly because of the carrier proteins, because the carrier proteins get saturated in some point. So that is the very important reason for the saturation point. Here we need carrier proteins, and carrier proteins can get saturated. So this is facilitated and simple diffusion. So this is one type of the passive transport mechanism. The other type of passive transport mechanism is osmosis. So diffusion of water or any other solvent molecules through a semi-permeable membrane from a solution containing lower concentration of solutes towards the solution containing higher concentration of solutes is the definition for osmosis. How to understand this definition? It is very simple. Just with this diagram, you can remember to your life. Just remember, here you can have the solvent. Water is the solvent. Here we have semi-permeable membrane. Here we have solutes which are higher in concentration. This side there is no solutes, right? So just remember these points. It is very important. Here in this picture we are having the solutes which are sodium chloride. Sodium chloride is the solutes here. So here in this diagram you can see the solvent molecules move through the cell membrane from the area of concentration of lower concentration of solutes to the higher concentration of solutes, and this is called osmosis. So what happens? There is movement of water, that is the transport of or diffusion of water from the area of lower concentration of solutes towards the higher concentration of solutes through the semi-permeable membrane. So that is the definition for osmosis. And here you can see there is some pressure. When some pressure is applied on the side of higher concentration of solutes, this prevents the osmosis of water, and that is the pressure which is called as osmotic pressure. Normally the osmotic pressure is exerted by the colloids which are present in the fluids. So in our body, the osmotic pressure is normally exerted by plasma proteins which are present in the plasma of the blood, and this the colloidal osmotic pressure is mainly exerted by a plasma protein which is called as albumin, and the normal colloidal osmotic pressure is 25 millimeters of mercury. Just remember, this was the MCQ which was asked in previous year university exam. What is the normal colloidal osmotic pressure or oncotic pressure? We can say it is normally 25 millimeters of mercury. That is the normal osmotic pressure. And these are the terms which we must know. So osmol, osmolarity and osmolality. What is osmol? Number of osmotically active particles in a given solution. So osmotic pressure depends on this osmol, the number of osmotically active particles in a given solution. Osmolality, which is number of osmotically active particles per kilogram of a solution, and osmolarity is number of osmoles per liter of a solution. So what is the normal plasma osmolality? Normal plasma osmolality is 290 milliosmoles per kilogram. 290 milliosmoles per kilogram, and this was the MCQ which is asked in this year exams. So 290 milliosmoles per kilogram, that is the normal osmolarity. So that's all for this today's class, and we will move to the MCQ session. I think you will be able to answer. Just remember all those points, and you should be able to answer all these MCQs. Which of the following moves rapidly across the cell membrane? Carbon dioxide, water, glucose, and urea. So which of the substance will move rapidly across the cell membrane? The answer is carbon dioxide, because carbon dioxide being lipid soluble substances, carbon dioxide will be transported through simple diffusion. So it will move rapidly across the cell membrane. And the next MCQ is osmotic pressure of solution is related to number of particles dissolved in the solution, chemical composition of the solution, size and type of the particles, number of equivalents of an electrolyte in solution. So what is the answer? The answer is number of particles dissolved in the solution. So osmotic pressure depends on number of particles dissolved in the solution. Which are the following transport mechanism across the cell membrane can get saturated? Which substance can get saturated? So you know saturation point is present only in facilitated diffusion mechanism. Now you try to answer. Diffusion of oxygen, diffusion of glucose, diffusion of sodium, diffusion of water. So which substance can get saturated? Yes, it is obvious. Diffusion of glucose. Here we can also give diffusion of amino acids, because they are also transported by facilitated diffusion. So it's diffusion of glucose reaches saturation point. This is very important to understand. In diabetes mellitus patients, nowadays we are having more number of diabetes patients in our houses, and you know that in diabetic patients they will have their urine will present with the glucose, which means glycosuria. There is presence of glucose in the urine. This glycosuria is due to it is mainly because of the filtration of glucose. Not all the glucose can reabsorbed in the proximal convoluted tubule. The very important point we must remember is, we will read this detailedly when we study excretory system. Just remember the glomerulus, which means in the kidney we know the nephrons, which are the functional units of the excretory system, and in that nephrons we are having glomerulus. In the glomerulus filtration will occur. So any substance which is get filtered will reabsorb in the renal tubular portion, and if it is not wanted by our body, it will be secreted into the urine, so into the lumen, then it will pass through the urine. So these are the basic mechanisms which happens in the renal tubules. Whereas in the renal tubules, especially in the proximal convoluted tubule, 100% of glucose which is filtered through the glomerulus will be reabsorbed. But the main mechanism which is involved in the reabsorption of glucose is, it is mainly through facilitated diffusion. Whenever sodium is reabsorbed, glucose will also reabsorb through the along with the carrier protein. So glucose will also be reabsorbed in the proximal convoluted tubule. So you know facilitated diffusion will reach saturation point. So once the saturation point is attained, what will happen? All the remaining glucose molecules will appear in the urine. They will not be able to reabsorb into the blood. So this is very important concept. So glucose molecules will have the saturation point. Similarly, like amino acids. Gases such as oxygen and carbon dioxide cross the cell membrane by, you know this, it is mainly through passive diffusion. We didn't study about other type of transport mechanisms, that we will study in the next class. So it is not through active transport or secondary active transport. And gated ion channels open or close by alteration in membrane potential, only when they bind to internal ligand, only when they bind to external ligand, depending on the lipid solubility of an ion. So gate channels mainly depends on the membrane potential. When there is a change in the membrane potential, I told you there are voltage gated channels which mainly open or close based on their membrane charge, right? So whenever there is interior of the cell membrane is loses its negativity, voltage gated sodium channels will open. So it is mainly depends on the membrane potential. And there are some channels which are called as ligand gated channels, right? So it is not only when they bind to internal ligand, or it is not only when they bind to external ligand. They can bind either with internal ligand or with external ligand. So this is not the answer. So whenever there is change in the membrane potential, these gated ion channels will open or close. So what is the normal plasma osmolality? It is 300 milliosmol per kilogram. So I told you 290. The range is 290 to 300 milliosmol. So here the answer is 300 milliosmol per kilogram. Rate of diffusion of a substance from a region of its higher concentration to a region of lower concentration is directly proportional to. So this is simple diffusion, right? This is passive transport. So substance should be transported from higher concentration to lower concentration. It is directly proportional with molecular size, temperature, thickness, and water solubility. So if the size of the molecule is large, the diffusion will be less. If the thickness of the membrane is more, then diffusion will be less. The water soluble substances will not have much rate of diffusion, but the temperature, I told you, if the temperature is directly proportional with the rate of diffusion. So the answer here is temperature. Normal oncotic pressure is 25 millimeters of mercury. I told you plasma protein exerts this normal osmotic or colloidal osmotic pressure, or we can also say oncotic pressure, which is normally 25 millimeters of mercury. So this is the answer, which is especially exerted by albumin, which is one type of plasma protein. So thank you. And one more thing which is very important is many of you are not downloading the notes which is given in the given with the link which you can open in the description. So if you click the description, you can open the link, and through the link you can go and get the notes. Please you make use of it, and please watch the entire video. Only then you will be able to understand or answer this MCQs. Thank you.